Good evening to all the BIPC second year students, and here we are to exercise the very short answer type of questions. Let's begin with the chapter Mineral Nutrition. So here, name any two essential elements and the deficiency diseases caused by them. Mineral nutrition is a very important topic for your Year BIPC exam because you can see multiple previous year questions, either match the following type of questions or statement based questions from this chapter, only on the essential, non-essential and toxic elements for plants. Okay, so if you look into two major essential elements that we have to focus, first is nitrogen, whose deficiency will lead to chlorosis and yellowing of the leaf. Okay, nitrogen deficiency will lead to chlorosis and yellowing of the leaf. And here you see phosphorus, the deficiency of phosphorus will cause stunted growth. It causes stunted growth and dark green leaves. Okay, both are macronutrients only which are vital for plant growth and metabolic activity. Next is define hydroponics. What is hydroponics? Multiple times asked question. You can see how many times it is asked. Hydroponics is a method of growing plants using nutrient rich soils. Using nutrient rich soils, the plants are being using nutrient rich solutions, plants are being grown without soil. Okay, and here the plant roots are supported in an inert medium like sand, gravel and perlite. It allows better control of nutrients. This allows better control of nutrient supply, and this will promote faster plant growth. This promotes faster plant growth. Clear? And then name two amino acids in which sulfur is present. Sulfur is present in two amino acids which we know very widely, cysteine and methionine. Okay, what are those two amino acids, ma? Cysteine and methionine. Cysteine and methionine are two sulfur containing amino acids. What are the vital roles of these amino acids? They are in protein structures and they also conduct the enzymatic activity. Sulfur will contribute to disulfide bond formation. They will contribute to disulfide bond formation and they will stabilize the protein structures. In disulfide bond formation and stabilizing protein structure, this amino acids, sulfur containing amino acids are being used. Name the essential elements present in nitrogenase enzyme. Essential elements present in nitrogenase enzyme. What type of essential elements are they? What type of essential elements you see? First, nitrogenase enzyme will contain molybdenum and iron as essential elements. They will contain molybdenum and iron as the essential elements, and these are categorized as essential micronutrients. Okay, they are categorized as essential micronutrients and they are crucial cofactors. They are required for enzymatic fixation of nitrogen. Fine, right? The nitrogenase enzyme, one more important feature. If you want to write for nitrogenase enzyme will be functional only in the anoxygenic conditions. Only in anoxygenic conditions. This point also you can mention out. Oxygenic conditions will make this enzyme inactive. And what are the elements present over here to be highlighted? Molybdenum and iron. Fine, right? Both are under micronutrients category. Next, explain the role of pink color of leg hemoglobin. Okay, pink color in the root nodules of leguminous plants. The pink color is due to the presence of leg hemoglobin. Okay, leg hemoglobin will give pink color to the nodules of the roots. This leg hemoglobin, this will facilitate nitrogen fixation. This will facilitate the nitrogen fixation by protecting the nitrogenase enzyme from oxygen. As I told you in the previous question, connecting question, when what are the elements that are present in nitrogenase is asked. Okay, nitrogenase is functional in anoxygenic conditions. Clear, right? Nitrogenase is present in is active in anoxygenic conditions. This ensures efficient conversion of atmospheric nitrogen into ammonia. Okay, this will convert atmospheric nitrogen into ammonia for plant use. Next, which element is regarded as 17th essential element, and name a disease caused by its deficiency. What is the disease caused by its deficiency? Nickel is recognized as 17th essential element for plants. Okay, nickel is recognized as 17th essential element for plants, and it plays an important role in urea metabolism and in nitrogen assimilation as well. Whenever you are addressing the very short answer type of questions, highlight the statements, find right. Always you need to highlight the terms which are important for the answer. Deficiency will cause what disease? Mouse ear disease in pecan plants. Next, write the balanced equation of nitrogen fixation. The balanced chemical equation is this. Okay, for nitrogen fixation, high amount of energy is required, where 16 ATP is utilized to fix 1 nitrogen and 8 protons and electrons are being utilized, where dephosphorylation of ATP is done to 16 ADP and inorganic phosphate, and in the products ammonia is formed, 2NH3 is formed. This is the balanced chemical equation for nitrogen fixation, important for your AP set as well. It represents the reduction of atmospheric nitrogen to ammonia. The process requires ATP, and this occurs through nitrogenase enzyme activity. This is all about the mineral nutrition. And next chapter we are going to address is enzymes. What do the 4 digits of an enzyme code? What do 4 digits of an enzyme code indicate? What does that indicate? The first digit shows major class. First digit, for 4 digits will be present, right? 1, 2, 3 and 4 digits. First digit will indicate the class of the enzyme. For example, we have 6 classes of the enzyme. It belongs to oxidoreductases or transferases. As such, the first digit will indicate the class of the enzyme. Second and third digits will represent the subclasses. Second and third digits will represent subclasses. And fourth digit will identify the specific enzyme within that subclass. Fourth digit identifies the specific enzyme in that particular subclass. Clear, right? This is the enzyme code. Find right with 4 digits. Next, about apoenzyme and cofactor. About apoenzyme and cofactor, you have multiple EAP set and NEET previous year questions also. In the recent NEET 2025 question also, there is a question asked on apoenzyme and cofactor. So, there is nothing, an isolated preparation for IPE and NEET. Okay, nothing like that. The same concept that you have to learn. First you see apoenzyme, and here is the cofactor. The protein part of the enzyme that is inactive alone. Okay, what is apoenzyme? Protein part of the enzyme, but only protein part of the enzyme is inactive. The non-protein part of the enzyme, which is essential for the enzymatic activity, is known as cofactor. Okay, apoenzyme requires cofactor to become catalytically active. Usually cofactor can be a prosthetic group, coenzyme or a metal ion. Any one of this can be a cofactor. This will assist the enzymatic activity. Next, together with the cofactor, it forms a functional enzyme called, this is called as holoenzyme. Okay, apoenzyme along with cofactor, this forms a functional enzyme. That functional enzyme is addressed as a holoenzyme. Combines with an apoenzyme, cofactor will combine with apoenzyme and this forms a holoenzyme. Holoenzyme is the functional enzyme. Clear till here? Next, define Michael-Menton's constant or Michael's constant. Michael's constant, that is Km, okay, Michael's constant is substrate concentration. This is substrate concentration at which enzyme activity is half of the Vmax. Okay, enzyme activity will be half of the Vmax. It reflects the affinity of the enzyme for its substrate. Okay, and a lower substrate concentration indicates higher enzyme substrate affinity. Okay, lower substrate concentration will indicate high enzyme substrate affinity. Who proposed lock and key hypothesis? Who has proposed lock and key hypothesis and induced fit model? Who has proposed them, both lock and key hypothesis and induced fit model? M.L. Fisher is the scientist. In the year 1884, he has proposed lock and key hypothesis. He has proposed lock and key hypothesis for enzyme substrate binding. Next it is Daniel E. Koshland in the year 1973 has proposed induced fit model. He has proposed induced fit model explaining the enzyme flexibility. Lock and key hypothesis where the active sites of the enzyme are rigid, whereas in induced fit model the active site of the enzyme is flexible. Both models will describe enzyme specificity, both M.L. Fisher's model and Koshland model, be it lock and key hypothesis or induced fit model, both will describe the enzyme specificity and catalytic mechanism. Fine, clear till here? And then we will move on to photosynthesis in higher plants. Name the process which take place in grana and stroma regions of the chloroplast. In grana regions, if you see, light reactions will occur. Here you can also mention out the 3 steps that happen in the light reaction. First is absorption of light in the form of photons. In grana region you can say first is absorption of light in the form of photons. And next, in the thylakoid lumen, inside the grana thylakoid lumen, photolysis of water takes place. Photolysis of water takes place. And last is formation of assimilatory power, that is ATP and NADPH are formed. These are formed in stroma, not in grana region. These are formed towards stroma. Fine. In stroma region, dark reactions will occur where Kelvin cycle, C3 cycle is operated. This will synthesize starch and enzymes. Both regions will work together to complete the photosynthesis process. Next, define law of limiting factors, which is proposed by Blackman. This is asked in the year 2016. Okay, so the law states that when a process depends on multiple factors, its rate is limited by the factor which is to the shortest supply. In photosynthesis, light, carbon dioxide and temperature will be the limiting factors. Improving the limiting factor, this increases the overall reaction rate. Overall reaction rate will be increased. Law of limiting factor is very important. This is asked quite a times as statement based question in your NEET as well. Clear? Next, where does photolysis of water occurs? Photolysis of water occurs in the thylakoid lumen. Okay, photolysis of water, this occurs inside thylakoid lumen. What is its significance? So here you see photolysis of water occurs in the inner side of the thylakoid membrane. Inner side of the thylakoid membrane will have a complex called as oxygen evolving complex. Oxygen evolving complex. Okay, this has oxygen evolving complex on the inner side of the thylakoid membrane. This will generate protons, electrons and oxygen during light reaction. Oxygen released is essential for the survival of living organisms. Clear till here? And next will be respiration in plants. Major question on ATP synthase, that is on F0 F1 complex, important for your NEET and MCET as well. F0 F1 complex. What is the specific role of F0 F1 particles in respiration? Specific role of F0 F1 particles in respiration. Here you see F0, F1 particles are protein complexes. These are located on the inner mitochondrial membrane. It will be very good if you can draw the diagram and explain as well. So here if you see, if you take a mitochondria, let us take a structure like this. Yeah, here this will be outer mitochondrial membrane and this inner mitochondrial membrane. This will be mitochondrial matrix. Fine, right? Outer mitochondrial membrane, inner mitochondrial membrane, mitochondrial matrix, and this is intermembrane space. F0 F1 complex is also addressed as complex 5, which is ATP synthase, where the complex is directed from the intermembrane space towards the matrix, where F0 will be inside the inner mitochondrial membrane and F1 will be towards the mitochondrial matrix. Here you see F0, F1 particles are the protein complexes. These are located on the inner mitochondrial membrane. Okay, where here F1 head piece will face towards the matrix, and this is responsible for ATP synthesis. ATP is formed inside the mitochondrial matrix in this way. The F0 base forms a proton channel. This allows the proton movement across the ATP gradient. Okay, that is from intermembrane space to mitochondrial matrix. Clear? Next, what is amphibolic pathway? Amphibolic pathways, they will function as both anabolic pathways and catabolic pathways. In both ways it will perform. Anabolic, which is building up, and catabolic, that is breaking down. They integrate energy production with biosynthetic activities. Okay, they will integrate the production with biosynthetic activities. For example, the respiratory pathway will act as amphibolic pathway here. Clear? Any doubt anywhere? Yeah. Next. Plant growth and development. Define plasticity. Last chapter from the plant physiology unit. Here you see, define plasticity and give an example, asked in the previous, like May 2018. So here you see, plasticity is the ability of the plant to modify its structure at different life stages. It allows plants to adapt to both morphological and environmental conditions. To both the conditions, the adaptation can be seen. For example, coriander leaves will show heterophily. Coriander leaves which shows heterophily is based on the phases of life. Different leaf structures are observed during the juvenile phase and different leaf structures are observed during the reproductive phase. So, coriander leaves will show heterophily, which is different leaf shapes in juvenile and in the adult stages. Next, what is meant by bolting? What is meant by bolting and which hormone causes bolting? It is of course gibberellic acid. Bolting is sudden elongation of the internode or shoot before flowering. Gibberellins, one of the best application of gibberellins that we have learnt. Spraying of gibberellins prior to flowering will increase the internodal length and increase the length of the floral stalk, whereby post fertilization you can see enlarged vegetables, fruits and all. This is the best application for rosette habitat. It commonly occurs in the plants with rosette habit such as beetroot and cabbage. Okay, the hormone gibberellins will induce bolting here. Clear? Next. What do you understand by vernalization? What do you understand by vernalization? Vernalization is nothing but this is an induction of early flowering when they are exposed to low temperatures. This is induction of early flowering when they are exposed to low temperatures. It ensures plants flower at proper season, typically after winter. For example, winter varieties of crops like wheat and vernalized to flower in spring season. Excuse. This is one of the application. Next, what is apical dominance? Name the growth hormone that causes apical dominance. Apical dominance is the suppression of lateral bud stage. Okay, this is the suppression of lateral bud growth by the activity of apical bud. It helps to maintain upward growth of the main shoot. The hormone auxin is responsible for causing what? Apical dominance. This will cause apical dominance. Clear? Next, define respiratory climacteric and name the plant growth regulator associated with respiratory climacteric. Of course, it will be ethylene, right? It is ethylene. So, respiration here, respiratory climacteric will refer to rise in respiration rate during the time of fruit ripening. During the time of fruit ripening, rise in the respiration rate is addressed as respiratory climacteric. This is crucial phase in the maturation process of climacteric fruits. The plant growth regulator is associated with it is ethylene. You can begin the answer with ethylene also, because for every 2 marker questions, that is VSAQ, you have to give straight and prompt answers. Okay, where answers should be very accurate. Okay, so this is about plant physiology. In the coming session, we will address the very short answer type of questions from few more chapters, which will accelerate your preparation. Till then, revise more and more, practice more. Don't learn in a way like IPE preparation is separate and your MSET NEET preparation is separate. Nothing like that. The entire concept is same itself, but here you have to learn the objective applications. Here you have to learn the subjective pattern of answering. That's it. Okay, develop that pattern of learning than learning it isolatedly, which will reduce your tension. Okay, and it will ease your preparation. Okay, so that's all for today's session, ma. If you are having any doubts and if you require any more tips for the preparation, you can drop messages in the comment section. We are here to address it.